Hi, we are here at the ASPB Plant Biology poster session, and right now I'm at the poster presented by Doctors Ping He and Li Boshan's group. They are the plant innate immunity signaling group at Texas A&M, working in Department of Biochemistry and Biophysics and plant pathology and microbiology. So I would ask Dr. Ping He to tell us who is here today in your group and what is the poster about. And maybe if you're a graduate student and want to talk about it,
Okay.
we will please first introduce your project and also who are in your group.
Okay, so actually I'm Ping He. So our lab mainly uses biochemical and genetic approaches to understand how plant respond to pathogen attack. And this is one poster presented by student use genetic approach to identify components in plant immune signaling. Okay, and so this is Chen, a graduate student, and Fang Jun, another graduate student, and Fu Hao, another graduate student, and Kevin used to be an undergraduate in the lab, IU student, and Bo Li, postdoc, and web, and I think it's okay.
Okay, and that's Dr. Li Boshan.
Yeah, hi. Welcome to Texas. I'm Li Boshan from plant pathology and microbiology. Similar, we were intrigued by the fact actually plants live in a very harsh environment, but most time they are healthy. And so we use genetic and biochemical approach to dissecting the immune signaling transduction pathway. I think I passed to the graduate student Chen Chen to explain his poster with you.
Hi, I'm Cheng Zheng. I'm also an Aggie PhD candidate, and so our work is mainly work on plant innate immunity, and we identified some mutants, and we named these mutants as Arabidopsis gene governing immune gene expression mutant. So this gene we called Aggie. So this Aggie 126 has enhanced immune gene promoter activity as here Aggie 126, and we focused on Aggie 1 for the more biological analysis. So here the Aggie 1 mutant can respond to different pathogen elicitators such as FLAG22 or F18, and also can respond to the different pathogen and have elevated FRK1 promoter activity. And also with that the disease assay to this mutant, Aggie 1 is more resistant to the virulent pathogen compared with the wild type. So this indicates that Aggie 1 is a negative regulator of the innate immune signaling. And we did more biochemical analysis, and we tested the MAP kinase activation in Aggie 1 mutant, and there's no detectable difference. This indicates that Aggie 1 is downstream of MAP kinase cascade or independent of MAP kinase cascade. Similar for the LS production, Aggie 1 shows no detectable difference between wild type. So it's either independent or downstream of ROS production. And then we found out the Aggie 1 gene by using MAP-based cloning, and Aggie 1 is on chromosome 2 in a gene called CPL3. Aggie 1 is a truncated protein, lost the catalytic domain of the CPL3. So we complement Aggie 1 mutant with the full-length CPL3. And as shown here, the complementation line has wild type level FRK1 promoter activity, and also in the Aggie 1 CPL3-4 knockout mutants, the endogenous FRK1 expression was enhanced compared with the wild type at 0.5 hours. And also CPL3-3 and CPL3-4 two knockout line is more resistant compared with the wild type. So this indicates that CPL3 is indeed the Aggie 1 gene. And also Aggie 1 can dephosphorate the serine 2 of R polymerase 2 CTD. Here, as the in vitro assay, we construct 3 different versions of CPL3: N-terminal, C-terminal, and FCP domain. CPL3C terminal can specifically dephosphorate iron polymerase 2 CTD at serine 2 residue. So we validated this result in vivo assay. Here we use Y-type and Aggie 1 seedling treated with FLAG22, and we can see in Aggie 1 mutant the serine 2 phosphorylation was enhanced compared with Y-type, but not serine 5. And also interestingly, after FLAG22 treatment, the phosphorylation of iron polymerase 2 was enhanced in both Y-type and Aggie 1 seedling. So this indicates that phosphorylation of iron polymerase 2 was involved and regulated in the plant innate immunity. So, here's the model of the role of Aggie 1 in plant immunity. When the pathogen infects the plant cell, the flagellin can be perceived by the receptor called FL2 and transduce the signal to the downstream signal protein kinase cascade, such as MET kinase and CDPK. Transcription factors can be activated by these protein kinases to promote the immune gene transcription. Meanwhile, in this PTI pathway, the iron polymerase 2 CDD will be further phosphorylated, properly facilitate iron polymerase 2's transcription capacity, and the CPL3 or Aggie 1 can dephosphorylate CDD of iron polymerase 2, function as a balancer or negative regulator to attenuate immune genes transcription.
Okay, that's great. That's awesome work, and the more appropriate title would be maybe Aggie immune signaling, innate immune signaling.
Yeah,
right?
Aggie.
All right, thanks.